Thank you so much, Helen. Can you all hear me okay?
Yes.
Great. I'm happy Robin and Helen set the stage a little so I can go through a few pieces a little faster. So I'm Connie Mitchell. Thanks for that introduction, Helen. Michelle Embry is also on the line, who co-manages this project. But today I want to tell you about the Botanical Safety Consortium. And just a little bit, Michelle and I work for the Health and Environmental Sciences Institute. I'm not going to go too much in detail today, and our website is there. But really, we want to engage from scientists from the different sectors, so academia, government, and industry, and other nonprofits, to try and identify and resolve global and environmental issues. And we work through a few different mechanisms that you're going to see in the Botanical Safety Consortium as well. So for a lot of projects, we come up with frameworks. We might work on data sharing of data that already exist. We can perform novel experimental studies with our collaborators. We write manuscripts, we can develop tools, and we can attend or hold scientific meetings or trainings. So to jump in, and like I said, Robin and Helen and others did a great job setting the stage, but botanicals are very complex and they create a lot of chemicals. So there's a lot of different plants in use, and their secondary metabolites or the different chemicals that they don't use for growth or just regular use can exhibit a broad range of bioactivities. And as humans, we've been able to use these plants for a long time for different things like pesticides, pharmaceuticals, poisons, etc. And Robin also touched on this, but just to show that going from the plant material itself, there can be many differences in chemistry, and that can be due to things like climate or growing conditions or what else is around. And also the extraction and processing processes can change the chemical constituents in the plant. So what solvent you use, if there's any adulterations or contaminations. And also, especially in the US, a lot of times finished products are combined with other products as well, other botanicals or other things. And this can all change how humans are exposed to it, similar to with regular toxicology, based on the exposure duration, the genetic age or background of the person, and any pre-existing conditions. And again, especially in the US and a lot of times around the world, testing schemes are built on single chemicals for toxicity. So here I just highlighting the differences between drugs and botanicals. So for pharmaceuticals, a lot of times the regulatory structure is aimed at ensuring safety. And a lot of times they're assumed harmful until you prove otherwise. Compared to botanicals, especially, they're typically simple and consistent, usually just the active ingredient. And the biological activity is usually associated with the constituent. For botanicals, it's really different. A lot of times the regulatory structure is aimed at ensuring access, and they're assumed safe until proven harmful. There's that idea that since they're from nature, they must be safe. Like I've tried to emphasize, they're complex and variable. And sometimes the biological activity is associated with the whole mixture, or we don't know what's driving it. So that leads to some problems. So currently there's not many methods that we know if they work for botanicals as complex mixtures. And there's not a step-by-step guide to how you would test a botanical. A lot of times there's reliance on history of use data, which can provide great information, but it doesn't always capture longer-term effects like carcinogenesis. And a lot of times too, if you do need to perform a test, it's done in mammals, so like rats or rodents. And this is costly, and there's ethical concerns. And also, as we talked about, if the chemistry is changing, it might not be representative. There's also a lack of global consensus, and what each global requirement, which each global regulatory body requires is often different. And there's not a lot of existing toxicity data. So basically, with all these problems, a group of stakeholders, multi-sector, came together to form the Botanical Safety Consortium. So you see a paper here actually from Amy Rowe, who's going to be talking in a little bit about the formation. And we actually got the US Food and Drug Administration to announce the formation of it and give support. And we really want this to be a public-private partnership with folks from the different sectors, from government, academia, industry, and nonprofits, to advance the safety of botanical ingredients. And just that was in early 2019, and later in 2019, everything officially started. And this is again through the US Food and Drug Administration and then the National Institutes of Health in the US. And they named my institute as the convener. And we've been working regularly since the beginning of 2020. And our current charge is to evaluate the suitability of assays for botanicals as complex mixtures. So said another way, what methods can be used to better evaluate the safety of botanicals? And where we are right now is on the left side. So we don't know what new approach methods can be used to evaluate botanical safety. And where we want to be in a few steps and what we're working towards is having a framework and a toolkit to evaluate botanical safety. And just to note, when I say new approach method, what I mean is in vitro, in silico, or non-protected animals, such as zebrafish embryos or C. elegans, that can be used. I do not mean rats or mice or other mammal studies. So these are our different working groups. They were chosen because a lot of times these are the targets of botanical toxicity, on the left at least. And then we also have other considerations. So if we're looking at things in cells or in silico, we also have to think of how that's different than how a human's exposed. So there's going to be considerations of absorption, distribution, metabolism, and excretion. And also I'm going to talk a little bit later about botanical drug interactions, which is an issue as well. We have chemists involved and data analytics people, and we also have a great group of pharmacognosists who have really been helpful and cut across all the different groups and giving us that background information on botanicals. All right, so again, our end goal is we want to be to develop that framework and to have tools that people could follow if they have a new botanical. So to get there, we have our different working groups, and what they've done over the past year and a half, 2 years, is identify botanicals that have known toxicity or known safety based on just reported studies already, or animals or human data. And what we've done is we've obtained large amounts, and what we're working on right now is looking to see what constituents are in the different botanicals and making sure there's nothing unusual, so that if we see toxicity in our assays, it'll be driven by the plants themselves. And right now, they're being stored with one of our collaborators at the National Toxicology Program at NIH, and they're going to soon be shipped to assay labs and already have been in some cases. In parallel, the different working groups were selecting assays that already work for single chemical toxicity testing that could be used for botanicals, or we think could be used. And so we worked with both chemists and the data analysis to try and design the studies. And once that's all ready, we're going to start running the botanicals through the assays, really to see if the assays are working. So that's what we're focused on and what I want to emphasize. We're not trying to say if a certain plant's safe. We're trying to come up with the tools so you can say that. So what we'll do is we'll do some comparison to what's already there in the literature. And probably this will be a nonlinear process where we'll have to go back, see what's working. But again, in the end, we want to come up with a framework that people could use. And as we go along, we also want input from our stakeholders. I just have this slide here on what we're not doing, because I think I told you a lot about what we are doing, but just to be clear. So like I said, we're not trying to say a certain plant is safe. So for example, we're not trying to say ashwagandha is safe or ashwagandha is toxic. We're trying to come up with the tools that people could do that. Again, we're not, along the lines of that, we're not making regulatory decisions. We're not endorsing a particular product, and we're not taking things off the shelves. So what we've done is we've gotten large amounts from the actual either manufacturers or growers themselves of botanicals to test. And we're really interested in the mixture itself, not just the individual constituents. So we're testing the whole plant in our assays. Currently, we're focused on the oral route of exposure, but that could be something we expand once we have a working framework. And we're not performing mammalian studies. We're also not performing risk assessments right now. And we're trying to see what methods are working, but we're not the big V validation of trying it in many different labs and being very rigorous. Right now, we're just doing kind of a wide search to see what methods are promising. So I'm happy to share these slides. I know it's a lot of information and I don't have time to go through everything, but here are some of the assays we're looking at. Some have already been tried for botanicals, but we want to do it in a more systematic way. So for example, for gene tox, there's quite a few in vitro assays already existing. We have some that will provide information on multiple endpoints. So for example, zebrafish embryos, when you expose them, you can get information both on developmental and then also neuro and sometimes even other endpoints. And we have many related to liver toxicity, but a lot of these assays can be combined. One thing what we're trying to consider is cost. So we don't want to select really expensive assays and then people don't use them. So like I said, I can share this, or if there's specific questions, can go into details on these. And then this is another one where I don't have a ton of time, but these are the botanicals we've selected to start with. These were selected by the groups either for suspected toxicity or safety, or for sometimes for herbal drug interactions. And again, I want to emphasize the botanicals were selected to evaluate the assays. The focus is on the assays, not the botanicals. I just want to run through real quick how we foresee this happening. So for an example assay, we would have different treatments. So we would have individual chemical negative and positive controls. And then we would have the ones that we think are positive for botanicals and those that we think are negative for botanicals, and in some cases others. We would run them in, say, a cell assay. We would expose them at relevant concentrations in the appropriate duration. We would measure our endpoints. We would analyze the data within the assay itself, but then we would also compare to the existing data. So if we think the plant is hepatotoxic or liver toxic, we want to compare that to the existing data. If we don't see it in our assay, we know something might not be correct in the assay, or there might be something unusual going on, and we need to refine and retest as needed. And our ultimate goal, like I said, is to create this framework. So what could you do with a framework? So you could screen for new or data poor botanicals. You could take products on the markets to compare them. You could prioritize animal botanicals for animal testing. So, or say which ones do not need animal testing. And you could also fill knowledge gaps. And so just real quick, here's our timeline. And so we've been working since 2020. Really, it took some time to get all the groups together and form our mission and scope. And then through 2020 and 2021, we selected our botanical case studies and began to think about assays. And we were also obtaining the botanicals themselves. Early this year, we finalized our assays and we started our chemical analyses. We're about to begin our assays, so it's actually a very exciting time. And once we get the results from that, we'll do our data analysis and silico analyses as well, once we have the chemical analysis data, and continue testing. We want to make everything publicly available and open access, so we will publish our results, and we'll continue as needed and create that framework. So I do just have a few slides on global outreach. I know I'm close on time, so I'll go through these quickly. But we have our main focus at the framework, but we also have some partnerships with a college in Malawi. And it was formerly the College of Medicine in Malawi. It's now Kamuzu University. But we have really two really interesting projects that Helen's involved with, and along with some other consortium members. So I promise I'll be quick. So one is on a botanical-drug interaction project. And probably many people know in different parts of the world, there's both traditional and complementary medicine being used. But as non-communicable diseases are on the rise, like diabetes and hypertension, people are taking both conventional medicine and botanicals. And this could lead to botanical drug interactions. And there haven't been a lot of studies in Malawi, specifically for non-communicable diseases in botanicals. So we wanted to do a survey at one of the larger clinics they have there. And this was done with the collaborators in Malawi. We provided some funds and we've worked with them, but they've done all this work. So we created a study, a cross-sectional survey. It was done last year and we had about 300 patients that were adults taking one herbal. And when we collected different information and wanted to see what medicines and botanicals they're taking. So just real quick, this is the demographic. It was mostly women and mostly peri-urban, but it was a nice variety of ages. And what we found for medicines or traditional Western medicines is that metformin was the most common. That's a diabetes medicine, so that made sense. For botanicals, there was lots of different kinds. There's many that were common, but two we are focusing in because there's not a lot of data on them are the sausage tree and moringa. And we really want to see the co-occurrence of people taking drugs and herbs, drugs and drugs, and herbs and herbs. And what we found is most people are taking about 6 drugs and herbs combined. And we want to do deeper dives on a couple of these to see if there's any potential herbal drug interactions. And for the next piece that I'll go through quickly, I also want to highlight that 5 people were taking Aristolochia. And so our next steps are continue to dig in. We actually are getting an intern to help us work through this project and do literature searches to look for mechanisms. And we plan to publish this work with our group in Malawi. And the second piece is the Malawi Aristolochia project. And like I said, Aristolochia was found in some of the survey, and Aristolochia hackiae has been found to be taken by patients. And Aristolochia hackiae is used for diabetes and different things, but the constituents actually aren't known. And why that's a problem is other species of Aristolochia are known to lead to kidney failure, DNA damage, and other cancers. And so there's some examples on the slide. So there's been issues in Belgium with women who thought they were taking one plant and it was adulterated with Aristolochia fang-G, and it led to end-stage renal disease. And also in the Balkans, people have issues with wheat being contaminated with aristolochic acids. So this is really a problem, but we don't know if Aristolochia hackiae contains aristolochic acids or in what amounts. So we want to do the chemical analyses to see if it contains it. And we're looking at a couple different species of Aristolochia, including Aristolochia hackiae, and we plan to look at the chemical constituents. They've collected most of the plants. Aristolochia hackiae they couldn't find last fall, so they're supposed to be looking this time of year. And our next steps are to run the actual chemical analyses, see if there's aristolochic acids, and then if there are, try and develop an outreach and education program for the potential hazards. So I know that was a lot. I know I went over time. I really appreciate your attention. There's more info on our website. And you can also email myself and Michelle. These aren't closed groups. We're really always looking for more people and experts and different perspectives. So thank you so much. I really appreciate it.
Much, Connie, for that wonderful presentation. A lot of information about what BSc is doing. And I'm sure everybody will be looking to understand this more. And I've seen that a lot of people have asked for the presentation. We'll work with David to upload these available presentations at a drive where you can all then access it. I think that would be the best way to share it.
Great.
So I have been looking at the chat and I noted a few questions. We are going to go to break, but maybe we can handle one or two of these questions before. So Rose Hayeshi asked that regarding the things that you are not doing, is this something that you will look into in the future or partner about in the future?
Yeah, some of them I would say yes. So for example, the oral route is what we're focused on now, but I know a lot of people are interested in dermal. So if you expose things through the skin, so that's one we can expand to if we have the right people involved and there's interest in the need. Some of them no, so we don't plan to do the rodent test within the consortium. If that is needed, we would, maybe say we don't have the, with the tools we have, we can't say for sure if covering that mechanism, but we ourselves wouldn't do it. I don't know if Michelle, you want to add to that at all?
No, I don't think so. I think you covered it. I think we're open. And I just saw another comment on herb drug interactions in animal models, like for agricultural species. And that's something that I think if there's interest and a need for it, we can discuss within the consortium and take those projects on depending on, what other things that we have on our plate or other opportunities and partnerships. So
there you have it. They are very open to collaboration. And I see a question here. Does anyone need to be in the medical field to be part of this initiative? I'm a traditional healer and also skin therapist. Basically, I specialize in health and beauty. I am sure there'd be happy to have you as a collaborator. And anybody else who is interested, you're welcome to reach out to BSc, to Connie or Michelle.
Yes. Yeah.
Yeah,
I think we're
Helen,
having,
please. Yeah, sorry, Connie. Just please distribute our email addresses, and if you have questions, feel free to email Connie or myself directly, and we'd be happy to talk with you more.
Fantastic. Any other burning questions before we take a break to digest all this?
Yeah, so I was just very interested around Aristolochia, and I didn't realize that we had it around here, so because, we also tend to have very high levels of kidney disease in this part of the world. And I know that the Balkan nephrology issue has been floating around. Some people have said it's because of mycotoxins. Yeah, so I think it would be very interesting information that you come up with, to
I
see.
think that Michelle has a lot
Yes.
of expertise with aflatoxin as well. They have a project in a different committee, but also looking into that for the mycotoxin piece.
Yeah, and I think I have a few papers, David, I'm happy to share, where they did a review of the different Aristolochia species and looked to see whether Aristolochic acid was present. So I think there's a lot going on, but I think it's really interesting because there are so many different Aristolochia species, and some of them not well characterized. So
yeah, very important work. Thank you.
All right, great. We thought we'd give you about 5 minutes.
Sorry,
bio
before that, I think there's just Kopano has a hand up. Do you want to ask a quick question, Kopano, and then we will take a break?
Yes.
Hi, yes. So I'm not a doctor, but I'm somebody who uses a lot of natural botanicals for my own and my family's well-being. But of course, when you research and you're trying to understand, the benefits of different herbs, especially, you learn around this issue of toxicity. And I've asked, some people that I know to say, for example, do we have a measure if somebody says, for example, we got something called mohlonyana, which is, I think, Artemisia afra, and how much is too much and how much is not. And related to that question is when we look at historical data so we've been taking it throughout from my grandmother to my mother to our generation, and yeah, we have not died, and we have not.
Okay, no, that's great, Kopano. So let's do this. Let's take a break, and then maybe at the end you will find your answer.
Yes, I believe you'll find some answer.
Yeah.
Towards the end. So
all right.
Let's give it a break, 5 minutes. We come back at what time is it now?
3:05 for those of us.
5, yes.
9. Yeah, 5
yeah,
hour.
5 past.
Okay.
Yeah, 5 past the hour.
You can just stay on. We're just going to leave the room open. So see you now.